Good afternoon, everybody. I am Cor van de End. I am a pediatric pulmonologist from the University Medical Center in Utrecht, the Netherlands, and I have the honor to present you something about the European HCF project. This is a combined European initiative to bring CFTR modulators to CF patients with rare genotypes. We have a website of the HCF project, www.hcf.org. So if you have any questions with regard to this presentation or with regard to the project, or if you want to reach out for collaboration, please go to this website and we will definitely be in touch with another. These are my disclosures. My institution receives payments for clinical studies and ad board meetings from diverse pharmaceutical companies. And my department received payments for research from diverse institutions like patient organizations and the European Commission. As we all know, cystic fibrosis is a very heterogeneous genetic disease. About 3% of the patients have gating mutations. Over half of the patients have homozygous delta F mutations. About 10% of the patients have delta F mutations on one allele and a residual function mutation on the other, and about 22% of the patients in Europe have a delta F mutation on one allele and any other on the second allele. After that, there is a residual group of about 10 to 50% of the patients who have very diverse and very rare genotypes, often only couple of patients with the same genotype are available all over the world. Fortunately, there are a lot of CFTR modulators coming up also in Europe. We are very happy that we have the ivacaftor available for patients with the gating mutations. And elsewhere in the symposium, I will present the long-term outcomes of ivacaftor treatment in patients with gating mutations, and what you see is that there is a rapid increase in lung function after start with ivacaftor, but also that this increase persists over long-term follow-up. So these compounds are very promising for the long-term future. For patients with homozygous delta F mutations nowadays, combinations like lumacaftor, ivacaftor, or tezacaftor, ivacaftor are available. And recently in Europe, there was the introduction of tezacaftor also for the indication of patients with delta F mutation on one hand and a residual function on the other allele. Of course, we are all looking forward to the introduction of the triple compound of Vertex in Europe, the combination of tezacaftor, ivacaftor, and elexacaftor that looks to be very effective in these patients, but also afterwards there will be a remaining group of patients with residual mutations that cannot be treated by these available CFTR modulators. So the goal of the HCF project was to target these residual patients to leave nobody behind. The HCF project is funded by the European Commission and it started in January 2018. The goals of the project are depicted here. The first goal, of course, is getting drugs available for patients with the ultra-rare genotypes, but also to further validate the organoid model as a predictive tool for personalized treatment in patients with rare genotypes. And the third goal is to create a biobank of organoids for future use in the CF community. Here you can see the consortium of the HCF project. From the early beginning, it was a project driven by the clinical trial network of the European Cystic Fibrosis Society and also the European Patient Organization. CF Europe was there from the early beginning of the project and was one of the important initiators of the project. Several laboratories are collaborating within the project. laboratories of the University Medical Center Utrecht, but also the University of Lisbon, the University of Leuven are working together. And this is the hub. This is a laboratory in the Netherlands that invented the organoid model and is included as a reference lab for the others in this project. Several pharmaceutical companies collaborate in this project. I can mention ELOX and proteostasis therapeutics, and they bring in their compounds to be tested in the organoids of these patients. There are several supporting partners for carrying out the clinical studies that are foreseen in this project. The project consists of 2 stages. The first part is collecting biopsies of about 500 patients with ultra-rare mutations in Europe, and to subsequently test these organoids with the compounds of the participating pharmaceutical companies. Afterwards, we will select the best responders from these in vitro testings and bring them into a clinical trial to test also the clinical efficacy of these compounds in high responding patients. Well, for those who are not so used to the organoid model, it all starts with a rectal biopsy. And it sounds a bit weird, but it's not a painful procedure. It feels like taking the rectal temperature, and it's taking a piece of tissue as big as about a pinhead, and that's fully painful. From this rectal biopsy, we can isolate stem cells, which are located in the crypts of the intestinal epithelial tissue, and they can be grown up with certain compounds in the lab, and they grow up into what we call mini organs or mini intestines, what are actually the organoids. So they can grow up from stem cells in the tissue, and they can be cultured again and again. They become deeply frozen, and they can also be used to test drugs on. Here's what you see happening in these organoids. The epithelial lining of the original intestine is available in this organoid, and the CFTR is expressed on the apical membrane, so on the inside of the organoid. If you stimulate the CFTR channels with forskolin, they start secreting mucus into the lumen of the organoids, and the organoid will swell, as you can see here in patients with a delta F mutation where no CFTR is available. You see no swelling of the organoids. But if you treat them with, for example, in this case, lumacaftor, ivacaftor, there's restoration of the CFTR function, and then the organoids start secreting lumen to the inside of the organoid, and they swell. So what we actually measure is the swelling of the organoids after stimulation with certain compounds over time. So it's a stem cell culture. The advantage is that we only need a single sample, and that we culture more or less eternal materials, so that we can be cultured to huge amounts of tissue. They can be deep frozen, and they can be defrozen again, so they can be used over time again and again for many years while remaining genetically stable. They are very patient specific, so it's not only the CFTR function that is reflected by the patient, but it's the CFTR function within the genetic background of the specific patient. And a big advantage of the intestinal model over, for example, the respiratory model or the liver model is that the functional assay of the forskolin swelling is fully CFTR dependent. So it's not dependent on alternative chloride channels or other channels, but it's fully CFTR dependent swelling assay. Here is what we published, and also it was confirmed by others. that there is a nice correlation between organoid swelling in the laboratory, as you can see here on the x-axis, with certain compounds, and after treatment of the patients where the organoids come from, you see increase in lung function or no increase at all. Here depicted on the y-axis, and you see a nice correlation between the in vitro effects and the in vivo effects. The same goes, for example, for organoid swelling and sweat chloride decrease during treatment. Also here you see a nice correlation between these two parameters, in vitro and in vivo. And if you calculate the sensitivity and specificity and the predictive capacity of the model, you find a very nice predictive capacity of the organoid model for both the decrease in sweat chloride, but also for the increase in lung function. Although there's always variability also in the clinical endpoints, as you can imagine, of course. Well, where are we now with the project? We have started with collecting biopsies since about summer in 2018, and well, 47 participating CF centers from 16 different countries in Europe collaborated, and patients were recruited for biopsies. And nowadays we have organoids on stock of 502 patients from many different countries like Italy, Denmark, Poland, Portugal, Sweden, Czech, Spain, Israel, Belgium, France, and UK, and also, of course, Netherlands, Austria, Germany, Switzerland. So from many countries, patients with ultra-rare mutations are included nowadays. And we started testing the compounds of the pharmaceutical companies on these 502 organoids. Here you can see the distribution of the genotypes of the patients that are participating in the project. Here you see the patients per genotype. So there's about 30% of the patients who have one stop codon, and about 13% of the patients who have two nonsense or stop mutations, and about 56 patients have no stop mutations at all. So if we look to the division of the compounds that are screened, we can screen about 1/3 of the organoids with ELOX compounds, which are targeting nonsense mutations, and about 350 patients are being screened for PTI compounds who have both a corrector and a potentiator and an amplifier compound. And I give you the results of some intermediate screening results. Here you see a waterfall plot of part of the patients who were tested with the PTI potentiator corrector combination. And what you see that there is a huge amount of patients, over 50%, who have a response in the organoid swelling assay. So there is in vitro activity of the potentiator corrector combination of PTI in these patients, almost more than 50%. And of course, there are also patients who do not respond at all, but it seems very promising that we can find at least quite a couple of patients who can be treated with this combination of drugs in the HCF clinical trials. What is also important to see is that there is a big heterogeneity in the response to the different modulators. For example, here you see the response to PTI compounds, the corrector potentiator combination of PTI, and here you see the corrector potentiator combination of Vertex. And you see that there are patients who respond to the Tezaiva combination, but who do not respond to the PTI combination. But there are also patients who respond to the PTI combination, but who do not respond to the TES-IFA combination. So there is overlap, of course, between the responders and the non-responders of the both combinations, but there are also patients who respond to one combination and not to the other combination. And this is, of course, an argument for a personalized medicine approach as was adopted in this HCF project. Well, patients with in vitro response to drug will enter a placebo-controlled clinical trial early 2021. And on this slide, you can see the centers that will contribute to it. So there will be a clinical study with proteostasis compounds, and there will be a study with ELOX starting next coming periods. This is how the clinical study will look like. Of course, we have small numbers of patients. So we have designed a crossover study model with 26 patients per study. The crossover is an 8 weeks active treatment followed by an 8 weeks washout period and an 8 weeks placebo period, or randomly the other way around. So there will be a study with PTI compounds, and there will also be a study with ELOX compounds. And we will also have a study with patients who are not selected by the organoid model, just to validate the value of the organoid model in itself. Well, the third goal was to produce a biobank. And of course, we want to keep these organoids alive because they can be used for the future in the next coming years. Nowadays, we have these organoids for the HCF project, but we aim that they will also be available for scientific work and collaborations. And we can, of course, combine the CFTR functionality with clinical data of these patients, and we have meaning to collaborate with the CFTR2 initiative to get all these data available for the scientific community in the next coming years. And of course, the project and the organoid biobank can also help pharmaceutical partners to speed up the drug development pipelines, because you don't have to make the, for example, FRT cells with certain genetic genotypes, but you can derive them from the patient quite directly. So thank you for your attention. And again, I would like to refer you to the HCF.org website for all your questions and for asking your questions in the near future. So thank you very much to you for your attendance and to all my collaborators for this great enterprise. And we have done a great job so far, and we continue and will help the patients with these ultra-rare mutations. Thank you very much.
Thank you to the moderators for the opportunity to present in Symposium 27, Challenges of Precision Medicine in CF, on the topic of theratyping in clinical trials. My name is Marty Solomon. I'm an Associate Professor of Medicine at the University of Alabama at Birmingham. I'm also the Director of the Adult CF Clinical Center and an Associate Scientist specializing in clinical and translational research in the Gregory Fleming James CF Research Center. I would advise you to engage in the chat features, as well as email any questions or comments to me at gsolomon@uabmc. I have the following relationship related to this presentation to disclose, including grant funding from pharma and non-pharma sources, advisory board memberships, and consultancy. The general outline of this talk is to review the latest trends in biomarker development as it relates to potential theratyping trials, examine the conceptualization of potential types of trial designs, and review implementation and emerging data from these trials. Two examples of theratyping have motivated our study of this concept in rare mutations. and by extension, the concept of optimizing therapies for all patients with CF. The first of these is data from Eric Sorcer's lab at UAB and now at Emory, looking at P67L. In his group, looking at FRT and human bronchial epithelial cells, they noticed improvements of this formerly classified class 4 mutation of a conductance defect with increased CFTR activation in both these model systems to the combination lumacaftor-ivacaftor. This caused a mechanistic study in electrophysiologic undertaking, including whole and single cell patch clamp, which demonstrated that there is in fact a protein trafficking defect in addition to the poor conductance that was observed in these studies. Since P67L had already been reported in certain case series to have increased response just to ivacaftor alone, this motivated the concept of treatment with lumacaftor and ivacaftor as a potential theratyping example of enhanced optimization of CFTR activation of this potential mutation. In addition, the concept has been advanced in the premature termination codon W1282X. This protein has been known for some years to exhibit some trafficking to the cell surface that allows potential therapeutic benefit from potentiators and other protein trafficking and CFTR activating modulators. Data derived from a patient indicated that in fact ivacaftor therapy did increase the short circuit current in response to forskolin, but did not do this, in fact, in a comparison patient who had other premature termination codons like G542X. In a subject treated with ivacaftor who was W1282X homozygous, this patient had stability of both lung function and improvements of weight in the post-ivacaftor period compared to baseline, indicating some therapeutic benefit was being derived from this concept of a theratyped potential therapy for this patient. These types of studies and the desire to help all patients with CF has motivated recent ECFS and North American guideline type statements, which advocate the advancement of theratyping in several themes. First is that clinical trials for developing theratyping for expanded access to rare mutations and therapeutic optimization for all patients with CF may rely on surrogate endpoints or biomarkers in populations with rare mutations, and that optimization of patient-derived biomarkers is key for expanding access to modulators for rare mutations and enhanced understanding of the mechanism of the CFTR protein defect conferred by specific gene mutations to advance the most biologically plausible modulators for activation of CFTR in all patients. This general timeline of currently approved CFTR modulators is also motivating in the concept of expanding access for patients. As you can see, since 2012, there's been a steady uptick of the number of patients covered by approved modulators, especially in the adult and older children population. However, despite this overwhelming success, there still is about 15+% of patients who remain untreated or non-responsive to even elexacaftor, tezacaftor, ivacaftor, the most broad and expansively reaching of the CFTR modulator combinations. This means there's much work to be done to expand in rare populations and non-responsive mutations. And the first answer to this has been Vertex's press release indicating FDA acceptance of a supplemental no drug application for FDA-approved modulators for additional CFTR mutations, which appear to be ETI therapy responsive or other modulator therapy responsive in vitro cell data. The expanding landscape of modulators is helping to move along theratyping as well. Many are familiar with the concept of correctors and potentiators, which have been in development and have reached patient access in most recent years. In addition, there are other types of modulators that are being developed and modulator type therapies, including stabilizers, which prevent premature termination of defective protein at the cell surface, enhancers and amplifiers, which increase the amount of mRNA present by which to increase the amount of protein being translated, and furthermore, in correlation, in a corollary to modulators, there are premature termination codon suppressors and read-through agents, which cause the usual cell machinery to partially ignore a stop codon, which may enhance the amount of protein product, which can be then modulated by other modulator modalities which are either available or in development. One such example from proteostasis therapeutics is shown over here, the triple combination of a potentiator, corrector, and amplifier, which is under development, has reached phase 3 study, and we'll have actually a theratyping trial to correlate with this to optimize therapy for patients with F508del when multiple therapeutic options for this mutation are available. It's increasingly apparent that the development of effective biomarkers for advancing modulators is especially important for rare populations where large-scale randomized controlled trials, as has been the gold standard in the industry, cannot be conducted. Therefore, surrogate endpoints which may predict individual outcomes may help to advance modulators based on prediction of clinical response or enhancement of the clinical response to lend veracity of the activation of CFTR function as the etiology of improvements of clinical responses in patients with small populations like these types of mutations. Perhaps the holy grail of such types of biomarkers is the human bronchial epithelial cell, which has been advanced by Vertex Pharmaceuticals to move forward several different modulator combinations, as well as the initial development of ivacaftor. This conceptualization of CFTR activation in vitro cells has been expanded to increase the number of modulators which can be tested for preclinical assessment of modulator responsiveness in rare mutations. In this example, which led to the 2017 expansion of the ivacaftor label to additional mutations, FRT cells transfected with mutant rare CFTR DNA make stable cell lines with this mutant CFTR. These stable cells are then grown under polarizing conditions for use in CFTR functional assays. Examples given include the Using chamber experiment. And then the response to ivacaftor was mapped across the various libraries of FRT cells with rare mutations to define which of these mutations might be responsive to ivacaftor based on the in vitro response. This in vitro response was then used to expand the label, as I previously stated. This conceptualization has greatly expanded the interest in developing tissue sources by which to develop fully developed biomarker assay platforms. As you can see, there are a variety of tissue sources under investigation, which after derivation from a patient, can then undergo in vitro assays, including epithelial culturing and organoid development, gene and protein expression assays, and other in vitro cell properties, such as the aforementioned Using chamber experiments or forskolin-induced swelling assays, as is used in organoid cells, and then optimally can be correlated to clinical improvements observed with the CFTR modulator to either be an endpoint for a clinical trial of an N of 1 type design, or to define entry into various types of clinical trial designs for rare mutations. Extensive work has been done by the Utrecht to advance the concept of organoid swelling from rectal-derived tissues, and it's been shown that the amount of swelling in response to a modulator in vitro, in fact, in a group-wise fashion, does in fact correlate to improvements of lung function in these populations. The rare study has attempted to induce a collaboration between the CFFT lab and several large clinical trial sites, including our site at UAB, to advance increasing tissue for several genotype targets, which are listed here on the left, and collect several different shots on goal towards the optimal biomarker to predict a response to various therapeutics in rare mutations. So from these clinical sites, blood for iPSC derivation, nasal tissue for air-liquid interface cultures, and rectal biopsies are sent to the CFFT lab for the development of iPSCs and organoids, as well as planar cultures to test library compounds, as well as optimization of therapies for other mutations in these genotype targets. I would invite you to look at poster 391 to learn more about the progress on the side of the biomarker development from Martin Mensa. This data that has come from one of the rare subjects who is a homozyte, who is a complex heterozygote for G542X and R553X, shows that in correspondence to a non-viable read-through agent plus lumacaftor, ivacaftor, advanced read-through agents identified through a compound screening library using the rectal-derived organoids shows that, in fact, the combination of CFTR modulators plus effective read-through can, in fact, induce forceful-induced swelling, which correlates to CFTR activation in this subject. Jennifer Gimbalot at UAB has detailed recent work in human nasal epithelial organoid swelling and has shown that, in fact, these assays correlate to improvements of percent FEV1 in sweat chloride with modulator treatment and may actually delineate a modulator response and delineate responders versus non-responders. In this data, human nasal epithelial organoids were derived after fibroblast co-culture and showed correlation between planar culture, short circuit current measurements, and the area under the curve, quantifying the forceful induced swelling of the organoids in response to CFTR activation. She's able to show there was a treatment level response with the highest increase of forceful induced swelling or area under the curve in response to triple combination of elexacaftor, tezacaftor, ivacaftor. And then, in fact, the level of CFTR activation here shown as drug response in the bottom left two graphs correlates well to absolute change of FEV1 in a subset of patients and the improvement of sweat chloride, which allows for a responder analysis assay to be performed to define responder versus non-responder in this organoid system. Work from Dave Nichols' lab has indicated that, in fact, in a proof of concept study that short circuit current measurements in air-liquid interface or planar cultures using human nasal epithelial cells may correlate to improvements both in short and long-term improvements in FEV1 in a study of 7 patients receiving initial treatment with ivacaftor for G551D or R117H CFTR. Work from my lab from the GOAL and GOAL extension studies has begun to extend the concept of biomarkers to downstream effects of CFTR activation, including airway surface liquid depth improvements. And we have shown that, in fact, compared to vehicle control in vitro, human nasal epithelial cell planar cultures of air-liquid interface show improvements of airway surface liquid depth when treated with ivacaftor in patients with G551D or R117H. And this correlates in a modest fashion to the change of those individual patients' percent predicted FEV1. So with numerous biomarkers which have various properties of both the capacity to predict response and potentially optimize therapies for both rare and more common mutations, this brings us to the concept of how do we unify this into the conduct of clinical trials for patients, especially with rare mutations. The ECFS guideline statement we mentioned earlier delineated the conceptualization that the traditional randomized control trial would be ineffective for patients that have a small number of patients per genotype. Thereby, other concepts of trial design would have to be undertaken, including the concept of basket trials or the N-of-1 trial. I'll begin to break this down with the next couple of slides. There are several considerations in trial design. There are 2 options which immediately become available and in the forefront when thinking about designing trials and are being advanced in the current clinical trial efforts. Option 1 of this is in which you look at individual biomarker response to define the entry criteria to the study. So in this type of study, the biomarker is tested first, and then that leads to the patient either having direct access to drug or attempted acquisition of the drug or entry into a clinical study which would advance clinical endpoints for optimization of therapeutics. In another option of clinical trial design, theratyping concepts using biomarkers would better define phenotypes both clinically or in vitro and better delineate mutation characteristics, as was seen in the example of the P67L mutation mentioned earlier, to define entry into a study. Then patients would have acquisition of drug in the study and clinical endpoints would be either correlated or otherwise to the endpoints in the in vitro biomarkers. This is an example from our lab of using the biomarker first principle, in which the biomarker first is used to predict if the patient will have a clinical response. That biomarker prediction must be optimized, of course, as we've alluded to earlier, and then the patient would go into an on-off open label study to determine if there's a clinical response to then attempt access for the patient or to expand, to apply for FDA labeling of the drug. Another example is an organoids-based response system, which is being developed in 2 different studies in parallel. The first is the HIT-CF study, which looks at optimization of rectal organoids and then testing optimal medication therapies for patients with individual mutations. This will be delineated more fully in another symposium talk in this symposium. In addition, Proteostasis Therapeutics is sponsoring the quote choice study, which is using a very similar principle to optimize triple combinations between theirs and other competing triple combination modulators for F508 and other mutations by using the organoids to predict optimal response in those patients. Another example, which uses the opposite kind of approach, is being advanced by Eric Sorcher and our group in collaboration also with Brian Davis, in which patients who meet certain clinical inclusion criteria that basically defines a residual function phenotype of CFTR activation, CFTR function, are enrolled in the study into an open label study of elexacaftor, tezacaftor, ivacaftor therapy for 1 month. In this, patients are enrolled and observed for change in particular FEV1 compared to baseline with several secondary clinical endpoints. And then also patients have the derivation from blood and other sources of iPSCs in order to measure short circuit and other endpoint measurements to correlate with the clinical endpoints to define the optimal biomarker response in this type of phenotype. Enrollment of this trial is underway and is detailed more fully in poster 432 by Rachel Linneman. And I would invite you to review this. And we do hope now that COVID has lifted in some of our studies for opening for clinical trials, we'll be able to fully enroll this study in the coming months. The CF Foundation has supported the efforts of Theratyping very well with the recent establishment of the National Resources Center at the Cincinnati Children's Hospital for optimization of 2 models of CFTR function using human nasal epithelial cells, both in air-liquid interface cultures and spheroid or organoid-based cultures. This work led by John Brewington is detailed in both posters 375 and 397 for more detail. In this, John has been enrolling subjects with rare CFTR variants and no modulator access, and using strictly preclinical access to predict modulator response. He then provides feedback to the patient and or physician to utilize for data for insurance approval or expanded access, with the goal of optimally defining predictive translational capacity of these models, which is shown in the next slide, which John has been able to demonstrate modulator responders versus non-responders using a very robust modulator response evaluation system that's under development there. So as you can see from this data, there are numerous types of trials and many different attempts at shots on goal towards developing both optimal modulators as well as the use of optimal modulator combinations as well as the optimal biomarkers to predict these responses. And as we can see, this will be soon translated into clinical outcomes with ongoing clinical trials, which are just awaiting continued enrollment. So with that, I'll conclude that there's promise for new modulators expanding to all patients with CF. It's clear that biorepositories and biomarkers are essential for acquiring a critical mass of tissue to test new therapies for rare mutations and optimizing therapies on both ends of clinical trial designs. The optimization of best readouts and biomarker systems are undergoing extensive investigation, and that novel clinical trial designs compared with these biomarkers may help to move drugs forward for people with CF who have rare mutations and optimizing available therapies for patients with more common mutations in the future. I'll acknowledge the patient participants in several studies, my co-lead investigators, and the research coordinators of several large observational studies where this data has been derived, members of my lab and key mentors and collaborators across the country, and funding sources of NIH and CFF. And again, thank you. Please email with any questions.
Hello, and thank you for joining me today. I am Jennifer Gimbalot, Assistant Professor in the Division of Pulmonary and Sleep Medicine at the University of Alabama at Birmingham. I will monitor the chat throughout the conference. If you would like to connect via chat or the networking platform, please contact me. I have no relationship to disclose related to this presentation. So today I would like to talk to you about the role of pharmacology in drug response. The pharmacology of any given drug is an important contributor to drug response, including on the individual level. Differences in absorption, drug metabolism, and transport can influence the quantity of active drug at the target site, resulting in clinically observed changes in patients. Today, I will mostly talk about ivacaftor, as it was the first modulator approved and the one we probably know the most about. This schematic shows a stylized example of the epithelial cell. In panel A, there is no ivacaftor and thus no potentiation of CFTR. In panel B, ivacaftor diffuses into the cell and potentiates the chloride channel, in this case, in the presence of no active metabolism of the drug, represented by the broken CYP3A, 4, and 5, which is the system known to metabolize ivacaftor and other modulators. In panel C, an active system of metabolism results in the conversion of the active drug to less active and inactive metabolites. An example of the clinical consequence is shown here. This is from a patient who was steadily involved in improving on ivacaftor when the patient was exposed to rifampin, which ramps up CYP3A, 4, and 5 metabolism. The patient experienced her first severe exacerbation with rapidly worsening symptoms within days of starting rifampin, had reduced weight and FEV1, which did not resolve until the rifampin was stopped. So this metabolism can take place in many tissues, influencing both the systemic concentration and the concentration that reaches the target tissue. Here is a diagram of the liver cell and the epithelial cell. When ivacaftor is absorbed through the gut, it is metabolized in the liver by CYP3A, 4, and 5 enzymes into metabolites. In the lower panel, ivacaftor is delivered into the epithelial cell, where it potentiates the CFTR channel. It is also metabolized and clears from this cell, presumably by the CYP3A system, which is active in lung epithelia, although with lower expression levels of all family members. So the variation in systemic concentrations can be predominantly driven by the absorption and metabolism initially, whereas concentration in tissue are driven by protein binding, distribution into the tissues, metabolism, and clearance at the site of action. So the drug concentration, or in other words, the exposure of the drug, is dose dependent, as seen on this graph here. That is from the initial review of ivacaftor by the FDA. Over the most of the dose range tested, the exposure, as measured by the area under the concentration time curve in PK studies, is linear. Furthermore, there is a proportional response to the dose in clinical measures. For sweat chloride and lung function, the effect appears to plateau, as seen here from the initial clinical studies of ivacaftor in 2010. Whereas for nasal potential difference, another measure of CFTR activity, the effect appears to be more linear. Together, these findings suggest that the dose is proportional to exposure and to outcomes, which would also suggest that the exposure is similarly related to the outcomes. And this forms the basis of my hypothesis that the drug concentrations, either in plasma or tissue or both, may influence clinical response to the drug. And this is important because we know that response to ivacaftor is variable. Dose selection is challenging and based on group-wise evaluation. Individuals within the group may have room to improve response based on their own unique factors. So in the panel on the left, you can see data from an observational study of ivacaftor. Over the first 6 months of therapy, you see each line is an individual patient's lung function response after taking ivacaftor. While it is a complex outcome measure influenced by many things, you can clearly see that some participants have a very large response, whereas others, maybe about 25%, have suboptimal responses. You see that this effect is not limited to ivacaftor, nor double combination therapies, but also seen in the most recently approved triple combination therapy, which is considered to be highly effective. The red arrows point to those with responses of 5% or change in lung function or less, while the mean change in these studies was about 10 or 14%. While it includes 3 different compounds, it does also include ivacaftor, and we expect that the concentration of ivacaftor and the variation of it are likely to be similar for both. So when considering drug concentrations and response, you must think not only of the population as a whole, but also each individual. Each person will have a distinct profile of drug concentrations, as shown here, where each line is considered to be a different person's response. Starting with the first dose, the blood concentrations will rise and fall over time until they reach a final steady state concentration, which is where the patient will remain overall. The goal is to get each patient to a steady state that exceeds the minimal effective concentration, but does not reach minimum toxic concentrations. Of course, ivacaftor is considered to be quite safe and doesn't necessarily have a toxic concentration, but there is still the possibility of detrimental effects or adverse events at higher concentrations. So each patient will follow their own trajectory, and maintain the concentration within the optimal window is important for many drugs, such as ciprofloxacin or tacrolimus. So realizing that the pharmacology of ivacaftor may be an important contributor to drug response, given the clear relationships between the dose and both exposure and response, I searched the literature for more data. And the data linking exposure to response is sparse, but what we do have shows that ivacaftor is highly variable in people. And the very best data that we have, it comes from the initial exposure response studies that are shown here. So these are the exposure response relationships for ivacaftor from the initial studies submitted to the FDA. Along the x-axes are drug concentrations, and the y-axes are response variables, either sweat chloride or lung function. I have added green arrows to orient you to what direction the better response is for each outcome variable. So I added this red line at a point where we could consider the patient to have had an optimal clinical response. In the case of sweat chloride, more than 60 points decrease and over 0.4 liters for lung function on these graphs. So starting in these red boxes, these are the lowest concentration ranges from 0 to 500. And within this range is the lowest effective concentration that we think should be maintained, about 423 nanograms per milliliter. However, you can see that even patients with the lowest doses, 25 and 75 milligrams, achieved some pretty robust responses for both measures, as shown by the red arrows, even though they had lower blood concentrations. Now look at these red boxes. This is the highest concentration in the blood. There are no optimal responders in this group for sweat chloride, and only a couple for lung function. And now here are the green boxes. These are in the range that's most people. So most people are taking the 150 milligram dose. And also the response, I've bounded the box that shows a response, not just the best response in this case. And so within this group, you see not only the largest numbers of optimal responses, but also larger numbers of any response. And as I noted, most of these people that are shown here are the green and purple triangles, those who took the 150 milligram dose, which was ultimately approved. So it is clear that this is the right dose for most people, but just maybe not for everyone. We want to know, how can we pinpoint the right dose for an individual, not just the entire group? Once approved and released, we often learn new things about PK-PD relationships for different drugs, and this data can help us figure out what these things may be. These graphs also demonstrate the difficulty of dose selection. As you can see, there is a lot of overlap in concentration achieved by the different doses, but also overlap in variability in response. And my conclusion from this is that you can not only achieve great responses for lower doses and exposures, but increasing the exposure above a certain level might actually blunt the response. And further, the exposure response is not linear, as opposed to the dose exposure relationship I showed you earlier, and at least in part, the dose and outcome relationship, which are somewhat linear. And furthermore, the exposure response relationship is not easily predicted in real world clinical use, where the exposure variability is likely to be much higher than in these controlled trials. So overall, these different features make it an attractive target to consider drug monitoring for personalizing therapy. And therefore, we needed a way to obtain the detailed data that will help us understand these relationships. To start with, one of my mentors, Dr. Ed Acosta at UAB, we needed a robust and reproducible assay for ivacaftor, as well as the metabolites and other modulators. And at this time, we can measure these compounds in multiple body fluids, including plasma and tissue lysates. And then we designed a typical PK study with sampling frequently over 12 hours, controlling as best as possible the patient's adherence before the study day, the meals taken, and the time of ingestion. We also use nasal brushing as a surrogate to measure the epithelial concentrations of the drug. And first, we obtain typical concentration time curves for the participants. And as you can see, we see a lot of variability, with 2 participants having very high concentrations, higher than have been previously reported. At least one other report has shown concentrations that were not quite this high, but also much higher than clinical use. The remainder of the participants fall within a range that's similar to what I showed you earlier. And to help you understand the comparison to the manufacturer's data, I've created this table where when we take out the outliers, you can see that our values for a few of select parameters are very similar. Whereas when including all participants, they differ quite a bit with a clearly higher level of elevated concentrations and total drug exposure, as well as variability. So, as I mentioned before, the high concentrations may actually blunt the response. And there is some precedence and some mechanistic studies that support this concept, although the studies have been primarily in delta F508 homozygote cells. The first suggestion of this detrimental effect came in 2014, when chronic administration of 1 micromolar ivacaftor was found to destabilize the F508 protein in vitro. And in one of those studies, the concentration of ivacaftor was found to be very high in the cell culture after chronic administration, which I'll show you later. This finding was supported by 2 other studies that further suggested the effect may not be limited to F508del CFTR alone and may in fact occur in other mutations or possibly be related to the destabilization of the lipid rafts in which the CFTR is found. And at least one additional publication shown here at the bottom assessed the free fraction of ivacaftor, which is a protein-bound drug that greater than 99% and with a very low free fraction. In this study, one conclusion reached was that the concentrations used in these in vitro studies were probably not relevant in vivo. However, that does assume that only the free fraction is important for what reaches the tissue. If ivacaftor is easily offloaded into the tissues and accumulates in vivo at high concentration, which has been suggested by not only its physical characteristics and also studies in animals, the low free fraction might not limit the concentration that reaches the target cells. So to study this phenomenon more closely, we looked at first in CFBE cells, a cell line commonly used in CF research, and applied 1 micromolar ivacaftor for just 24 hours. We removed it, washed the cells, and then subsequently lysed the cells at different time points. And we found approximately 4 to 5 times higher concentration inside the cells after only 24 hours of exposure that also remained above 1 micromolar 3 days later. It took about 10 days before the cells were able to have low, nearly undetectable levels of the drug. And this phenomenon has been further explored by another group, the one I mentioned earlier, that showed the destabilization of F508del in response to chronic ivacaftor treatment. After this treatment, they found that the ivacaftor was accumulating at higher levels inside the cells as well. And in this case, it was a human bronchoepithelial cell model. Most recently, Dr. Charles Esther at UNC has looked at this more comprehensively. Applying 1 micromolar ivacaftor or lumacaftor or tezacaftor modulators over 14 days. And as you can see with the blue dots, that ivacaftor concentrations after 24 hours is very similar to both of the 2 prior studies that I showed you. And furthermore, that over many days of exposure, the concentration inside the cells continued to rise. Once the cell was removed at this point, once the ivacaftor was removed at this point, the cells continued to maintain that high level of concentration and weren't able to clear it. And this was limited to ivacaftor and not seen in lumacaftor or tezacaftor, which were easily cleared from the cells, suggesting that, at least in either monotherapy or combination therapies, that ivacaftor is the one that may be accumulating at high concentrations and may be responsible to the effects that I showed you earlier. The conclusion from this is that if cells in vitro can accumulate ivacaftor over time to levels that are much higher than they're initially exposed to, then the impact on at least F508del CFTR may be detrimental. But then the question arises, do concentrations in vivo get that high? There have not been direct measurements, although the answer in animals is yes. Data submitted as part of the new drug application, initially to the FDA, showed accumulation in tissue in animals, but data in humans is less available. So we have started to collect some data on this, using nasal brushings as a surrogate for epithelial concentrations. And here is our initial pilot study on intracellular concentrations that was published earlier this year. While it's a small sample size and using 3 different drug combinations, including ivacaftor monotherapy, we found highly variable intracellular concentrations in the cells, and also that the concentrations could actually get up to the 25 micromolar range. So taken together, this preliminary evidence does suggest that not only can plasma concentrations be very high in real-world use, but epithelial cells can also accumulate very high concentrations of the drug. And this does seem to occur in vivo, regardless of the combination therapy, with the exception of ivacaftor lumacaftor, which we won't have time to go into today, but does have different metabolism interactions that make concentrations lower in general. So is there any suggestion that these concentrations matter clinically? Well, we have started to look at that too. And here I'm showing you the absolute change in lung function at 5 years, which is the closest time point to determination of the concentrations, and also the rate of lung function change as determined by simple linear regression. There is a trend toward correlation with absolute change in lung function, as you can see here on the left, and a significant correlation in the rate of change on the right. I see similar significant trends with exacerbation rate after initiation of ivacaftor monotherapy and other lung function measures, although not for weight or BMI. So in summary, we do see that ivacaftor concentrations are variable in people and may impact clinical outcomes, although further real-world study is needed. These findings have implications for double and triple combination therapy as we continue to fine-tune the precision approaches in this field. Expansion of these studies to more patients, to combination therapy, and to take into account the metabolism and clearance of these drugs will help us better understand if concentrations should be monitored to improve care. I want to thank the many mentors, advisors, and staff, my institution, and the Cystic Fibrosis Foundation and the NIH. In particular, I want to thank the patients and their families who volunteer for these studies, without whom this work would not be possible. Thank you, and I hope to hear from you on the networking platform established by the conference.
Hello everyone, my name is Mina Ogawa from McKeon Stem Cell Institute, Toronto. Firstly, I'd like to thank the organizing committee for inviting me to present our work today. We have been working with researchers at the Hospital for Sick Children, Toronto, where the CFTR gene was discovered, and multidisciplinary teams are working on CF clinical care and research. I myself have been working on stem cell research, and today I'd like to talk about how stem cell research serves CF society. I don't have any conflict of interest to disclose. As all you know, CF is a multi-organ disease, and many studies have been performed to understand pathogenesis of CF by using primary and stem cells. However, primary tissues of internal organs, such as the liver and pancreas, are relatively inaccessible and have not been studied very well comparing to the nasal or intestinal tissues. Today, I will talk mainly about cystic fibrosis liver disease, which counts for the 3rd most common death of CF patients. In the liver, cholangiocytes, bile duct epithelial cells express CFTR, and these green tubes are target organs. When CFTR is dysfunctional, dehydrated bile acid harms the duct, results in biliary cirrhosis. However, clinical biomarkers that reflect disease manifestation of CF liver disease have not been well established yet, and clinical benefit of current CFTR drugs is still under investigation in patients suffering from CF liver disease. Our understanding of the pathogenesis of CF liver disease is limited due to the inaccessibility of the tissue, and not all CF animal models demonstrate the liver disease manifestation correctly. So clearly, our understanding of CF liver disease would benefit from an in vitro model that recapitulates the impact of CFTR mutations on bile duct development and function. What are induced pluripotent stem cells? iPS cells are produced from somatic cells, like skin or blood cells, by introducing 4 transcription factors, OCT3 and 4, SOX2, c-MYC, and KEL4. They are also called Yamanaka factors. Dr. Yamanaka got awarded the Nobel Prize in 2012 due to this discovery. Once human iPS cells are generated, they are able to propagate indefinitely and also differentiate to any type of cells of your interest. For me, liver and pancreas are the target cells to study cystic fibrosis. What is the utility of iPS cells and how do they serve in clinic? Once cells are created, stem cells have been used for the drug discovery and profiling, which will be shown later. Currently, stem cell-derived neuron cells or retinal cells have been used for cell therapy in the clinical trials to treat Parkinson's disease or age-related macular degeneration, respectively. With the combination of CRISPR technology, many gene therapy strategies have been developed, which you might hear at a different session at this conference. Stem cells have a tremendous opportunity to tackle diseases. We have previously published a cholangiocyte differentiation protocol from human ES iPS cells. We confirmed human stem cell-derived cholangiocyte expressed CFTR proteins and primary cilia, which are the hallmarks of mature cholangiocyte. We modeled cystic fibrosis liver disease. Although successful, some limitations exist. First, direct CFTR measurement is difficult since CFTR was expressed in the apical side of the lumen. Next, quantification and functional assessment of primary cilia were not achieved. And finally, the low cell yield of 3D organoids were not amenable for high-throughput drug screening. These limitations motivated us to generate a more efficient protocol of stem cell-derived cholangiocyte, which are applicable for CFTR drug screening. How do we differentiate stem cells to cholangiocyte in a dish? We refer the knowledge of the developmental biology and recapitulate the developmental stages step by step. To introduce cholangiocyte lineage, firstly, we generate definitive endoderm, followed by hepatoblast, which is the bipotential progenitor, giving rise to both hepatocyte and cholangiocyte. We further mature cholangiocyte, which possesses primary cilia. We used both human embryonic stem cells and iPS cells for cholangiocyte differentiation. Developmentally, it is well known that notch signaling plays an important role for the specification of bile duct fate, and we mimic that by using OP9 stromal cells, which was engineered to express notch ligands. At the end of each stage, we confirmed the specific protein expressions. Efficient definitive endoderm was confirmed by the expression of CXCL4 c-KIT. Hepatoblast, which is marked by albumin and alpha-fetoprotein. At day 49 of differentiation, strong glycosylated CFTR protein expression was confirmed by Western blotting. At the differentiation day 49, cholangiocyte-relevant proteins were confirmed by immunohistochemistry. CFTR, shown in green, is expressed along with cholangiocyte duct marker cytokeratin 7. SOX9 is a duct transcriptional marker, and ASBT is the bile acid transporter. The left panel shows the higher magnification of primary cilia, and the video displayed on the right shows the abundance of primary cilia expression. Almost over 80% of cells demonstrate primary cilia. Why is primary cilia so important in cholangiocytes? In the liver, hepatocytes do not possess primary cilia, whereas cholangiocytes possesses primary cilia, and these small organelles serve as mechano, osmo, and chemosensor to control fluid secretion, maintaining bile duct homeostasis. Scanning electron microscopy shows here that 2 cells and each cell have primary cilia along with microvilli. They look very similar as seen in the primary bile duct. The red arrow shows the tip of primary cilia. After confirming hallmarks of mature cholangiocyte in the culture, we decided to investigate the utility of our stem cell-derived cholangiocyte. Cholangiocyte CFTR function is measured by membrane potential dye stimulated by first screen and the plate reader. To perform high-throughput drug screening, firstly, we examined Z-score in 96 well plate. To simplify, this measures the standard deviations below or above the population mean. As shown in the left bottom, the Z-score ranged in 0.5 to 1, which means it is an excellent assay, and our cholangiocyte scored 0.63, indicating they are good for drug screening. Here, I would like to introduce the CFIT program at SickKids Toronto, where over 100 patients have been recruited to develop cell-based and genetic-based assays to help predict who will respond to CFTR drugs. The idea is we differentiate bile duct epithelial cells from CF patient iPSCs and perform drug profiling to see if there is any difference among patients in response to CFTR drugs. Other research groups at SickKids have been differentiating CF patient iPSCs to the different organ cell types. So we will compare the response from the same individual in the near future. The plate reader cell-based assay has been developed by Dr. Christine Baer's lab to measure the CFTR function. These are the representative traces showing CFTR function in CF patients' derived cholangiocytes. This slide shows the quantification of the drug response. Here, drug efficacy is ranked by the direct CFTR measurement. When you look at the red bar, this is a vertex triple combination, exhibits better response than green, which is vertex 809 and 770 in both CF patient iPSC-derived cholangiocyte. Interestingly, the black bar, which is a triple combination from the other company, exhibits a different response in 2 patients, and we might be able to say that we can predict which drugs are beneficial to each individual. While the plate reader assay measures direct CFTR function, we wonder, will the cholangiocyte reflect indirect fluid secretion in the dish? To do so, we generated 3D organoids from monolayer cholangiocyte and performed the swirling assay. If CFTR is functional in 3D organoids, it regulates the fluid transport and the cyst size will be enlarged upon the stimulation of first screen. For CF patient-derived cysts, we expect 3D organoids require drug treatment to restore the swelling function. This video shows the swelling assay result from wild-type embryonic stem cell-derived cholangiocyte. As you can see here, cholangiocyte cyst swelled after the stimulation of first screen. This is the quantification of swelling assay. The left panel shows wild-type swelling response, which was blocked by the CFTR specific inhibitor. The middle and right panel showed the response of 2 delta 508 patients. Without any drug treatment, shown in gray dots, CF-derived cholangiocytes did not show any fluid secretion. The level of the rescue is not so significant. However, CFTR drug treatment restored the fluid secretion in patient-derived cholangiocyte cysts, which is shown in black dots. The last part of my cholangiocyte talk is about primary cilia. As I mentioned before, primary cilia plays an important role to regulate bile duct homeostasis. Rodent studies have shown that the mechanical stimulation of bile flow to transduct extracellular signaling and activates calcium signaling in cholangiocytes. To investigate the relationship between primary cilia and calcium signaling in human stem cells, we took advantage of GCAMP cells. GCAMP is a genetically encoded calcium indicator. With this, calcium activity is easily monitored by GFP signaling. To detect calcium signaling in cholangiocyte, we differentiated GCAMP cells. Firstly, we confirmed the flow-bent primary cilia. And as you can see here in the video, when we introduce the flow, it induces calcium signaling in GCAMP cholangiocyte. And this is a trace of its activity. Next, we wondered whether the mechanical flow in cholangiocyte affects CFTR function or not. We observed that the flow induces CFTR function. The function was amplified by a first screen and was blocked by a CFTR inhibitor. Interestingly, the CF patient-derived cholangiocyte did not respond to the flow stimulation, although flow-induced CFTR activity was restored when treated with CFTR drugs in CF patient-derived cholangiocyte, suggesting that defects in signaling-mediated primary cilia is involved in the onset of CF liver disease. I will wrap up my talk with the pancreas differentiation from iPS cells. With the same strategy, CF patient-derived pancreas cells were differentiated, and the hallmarks of each developmental stage were confirmed. Endoderm and pancreas progenitor were marked by NKX6.1 and PDX1. At the end of the differentiation, CFTR is highly enriched along with ductal markers SOX9 and cytokeratin-19. The CFTR function was measured after the CFTR drug treatment, and the Vertex triple combination drug showed the better efficacy than the 2 combination drugs, as what we observed in cholangiocyte from the same delta 508 patient. Strong protein expression was also confirmed by Western blotting. Let me summarize my talk. Cholangiocytes differentiated from stem cells showed CFTR function and can be used for CF liver disease modeling and drug predictions. Primary cilia-associated functional assay has established, and CFTR drugs rescued mechanical flow-induced CFTR activity in patient cholangiocytes. Pancreas cells are also differentiated from CF patient iPSCs and showed the similar trend in response to CFTR drugs. Future studies will reveal which type of functional assays will benefit to predict drug response in each affected organs when compared from the same individual iPSCs. Here is the acknowledgement, and I thank all people here and funding agencies. Thank you so much for your attention.